So I'm going to talk to you a little bit about Cell Blender and Blender and MCell and how to build a pretty complicated model with these tools to kind of get you inspired about what's possible with these tools. So I'm not going to walk you through a simple model building process in the pipeline, and then after lunch in the hands-on tutorial you'll start building much simpler models to get you going, and that's best done yourself, with your own hands on the tool without having me walk you through something like that. So I just want to give you an inspirational example of what's possible, and I was encouraged to hear that a lot of you have a neuroscience background and are interested in using MCell for doing neuroscience simulations, and what I'm going to show you is an example of just such a thing. But I also want those of you who aren't neuroscientists to not feel left out. Where it's possible to make all kinds of models with all kinds of biochemistry that can be from any kind of cell. And by the way, neurons are cells, right? So it's all cell biology. So Cell Blender is a tool that we've developed to make it easier to build these MCell models that Marcus has been showing you what the model description language looks like for. You can develop these applications in Cell Blender using the add-on called Cell Blender. It's an add-on to Blender. So what is Blender? And why are we using Blender? Blender is this amazing 3D computer graphics environment designed by computer graphics artists and computer graphics modelers to create three-dimensional objects and scenes for making computer animations for Hollywood kind of things. A lot of computer graphic artists felt left out by Hollywood because the tools that Hollywood has developed are very expensive. So a group of computer graphics artists and programmers got together and created their own open source version of some of the expensive things that Hollywood has to offer, and they were kind enough to make that available to the whole community in this tool called Blender. And it's pretty amazingly powerful. And it's a real change for the research community, the biology research community, and other communities, because doing things in 3 dimensions is really hard and complicated without a good tool to build these things. And over the years, a number of tools have been developed within the research community to do 3-dimensional things, but they lacked certain capabilities that are really hard to provide. And so the tools weren't very useful. And it would, these tools would get off the ground and go so far, and people would start using them. But then just the complexity of trying to produce a more useful thing was too big of a hurdle to get over. Until this Blender tool came along, and that community got together and finally said, we need to have this thing. And because of that, now we have it too. So, but before I show you what you can do with it for cell modeling, let me give you an idea of what can be done with Blender in the hands of the computer graphics artist community. So on blender.org you can find this video that just came out. It's 1.5 minutes. That'll give you, I think there might be sound. Oh. So this is an example of what can be done, and I want you to look at this and see some of the physics involved with what you see. So there's a lot of artistry here, but there's also a lot of just plain good Newtonian physics going on with some of the motions and interactions of okay. Oh, that's a good one right there. Yeah. So obviously you can put on a lot of bells and whistles and glitz here, right? But oftentimes what's visually compelling and artistic is also a very powerful way to communicate. That's one aspect of model building and computer simulation is the need to communicate what you've done with the audience that you're trying to reach. But there's a deeper message here, which is that in order to produce professional results like this, you really need a professional tool in order to do it and be productive doing it. And fortunately now, in the cell modeling community, we can use Blender in an equally productive way using that tool. And so that's what we're hoping to accomplish by making the Cell Blender add-on for Blender, so that now we in the cell modeling community can make use of this really powerful tool for our own purposes. And so what you saw were a lot of beautiful computer graphics, but underlying that there was also a lot of physics going on with the motions of some of the things that you saw. And in order to do that, they've built into Blender a physics engine called the Bullet physics engine, which is a Newtonian kind of physics engine. And with models in the future, with cell models in the future, we might make use of that Bullet Newtonian physics engine. But I want you to think of MCell as a statistical mechanics physics engine that we've plugged into Blender via the Cell Blender add-on. So really what we're doing is we're extending the capabilities of Blender with statistical mechanical physics that we use to do reaction diffusion simulations, but it's really an extension of the physics that we can access using Blender as the tool to create the worlds in which those physical processes happen. Okay. So what I'm going to show you now is making use of all that for creating a computer model of synaptic physiology. And our motivation for creating this tool was we're interested in this model is we're interested in how synapses work and how does synaptic structure influence synaptic function, and what is the role of synaptic plasticity in the neural code. That was the motivation that led us to create this this model that I'm going to show you. And what it's all about is this is stated very succinctly in this 1999 paper from Sains and Liquin, where they asked the question, can molecules explain long-term potentiation? So long-term potentiation is the process by which your synapses adjust their synaptic efficacy over time in response to the inputs and outputs of neurons at synapses. And what they asked the question, can molecules explain long-term potentiation? Of course, if we believe in the molecular basis of how biological systems work, and molecules have to explain everything, right, about how biology works. So the answer has to be yes. But they asked the question this way because what they realized is, what this community has been realizing is that these reaction diffusion systems, these biological, signaling pathways are so complicated that we can't make sense of how they work. The old days of when you'd be able to look at something and what the pieces were that made it up and how they interact with each other, and from those interactions you could do you could think about it, sit down and think about it and puzzle your way through it. You could figure out a hypothesis about what you thought was going on, and then you could design an experiment and go out and test this hypothesis. Those days are over, and because these systems are so complicated that you can't now sit down and figure out what all the possible interactions are and make sense of it by doing a simple thought experiment. And this then interferes with our ability to understand. Richard Feynman said very eloquently, "What I cannot create, I do not understand." And so our approach with using M-Cell and Cell Blender for modeling these things is we need to use computers as our allies in this process and achieve understanding through simulation. And another way to think about this is what I call the scientific discovery cycle that is enabled by scientific computing. When a system becomes so complicated that the normal cycle of making observations and thinking about the system and coming up with predictions and hypotheses that then lead to experiments, when the system is too complicated for this cycle to continue, then you're really stuck. And ultimately, the main problem is the problem of forming hypotheses, which is a mental process. It's done by thinking. Computers can't think of the hypothesis for you. All they can do is provide you, or your observations and the experiments you do can't provide you with the next hypothesis to come up with. That comes by thinking about what you've observed. And if what you've observed is too complicated to think about, then you can't make a new hypothesis and come up with new experiments. So you're jammed up, right? So ultimately, everything that happens in science is really a thought process, and you have to be able to do thought experiments on your mental model. Your observations and your experiments provide you with an intuition about the world. But if the world is too complicated to make sense of your observations and your experiments, then what you really need to do is a computational experiment. Put the components that you think exist in your model, put them into the computer, and let the computer sort out, with the laws of physics, and let the computer sort out all the interactions and provide you with results that, because it's a computational world, you can take it apart down to its microscopic components and probe it at the component level in your computer model, and then now all of a sudden you can start gaining new intuition about the system. And by doing computational experiments and forming new intuition about the behavior of the system, now all of a sudden you can start doing thought experiments again, because the computer simulations have helped to inform your thinking and improve your intuition about the system. And then you can go back to doing bench experiments again based on the new hypotheses that the computer simulation has helped you to formulate. So this is how we see the interaction between computational experiments, bench experiments, and thought experiments, and why, as we start to learn more and more about the deeper mysteries of biological systems and their complexity, why more and more we're going to need to rely on doing computational experiments and building more and more complex and realistic models. Okay, so if you're fortunate enough to have a system that's much simpler, you can make progress maybe without worrying about this so much. But at a certain point, though, you're gonna run into this intuition wall, and you're going to need to make more realistic treatments of your system. And so these are the kinds of systems that we're gearing our tools toward. An example of just such a system I'm about to show you, where I'm going to show you a model of synapses in hippocampal neural pill. And the simulation is going to include all of these components that have been studied over the years individually through various experiments. We've determined the resting level of calcium in the system, and the fact that what's involved are all these different pumps and voltage-gated calcium channels and NMDA receptors and AMPA receptors and release of neurotransmitter and calcium buffering and binding of calcium to these different target molecules, and more pumps and yet more pumps. And then on the presynaptic side, the mechanisms that are responsible for releasing the neurotransmitter. It's a dizzying array of all these things, and yet we believe that these are all the components necessary underlying the process where an action potential arrives at the presynaptic terminal, and calcium flows in and results in the release of a vesicle of neurotransmitter, and that transmitter diffuses in the synaptic cleft and activates receptors on the postsynaptic side, and then there might be an excitatory postsynaptic potential that results in activation of action potential, and that action potential back propagates up into the dendrite and activates voltage-gated calcium channels, and more calcium flows in. And it's a story that you can talk about, but until you put all the pieces together in the computer and see whether or not this cartoon story that I'm telling actually recapitulates the physical, quantitatively recapitulates the physical process that we can observe with microelectrodes and calcium imaging and these other things, how do you know that you've got everything put together correctly and that you really understand what's happening? So we set out to create just such a model from a 3D reconstruction of hippocampal neural pill. So in collaboration with Kristen Harris at UT Austin, we obtained a 3D reconstruction from her of a chunk of hippocampal neural pill. Here's a little slide. Let's see, do I have the pointer here so I'm not blocking the... Is there a pointer, Marcus? Oh, there's one in the remote control. Oh, yeah, that's good enough. So Kristen Harris took a chunk of area CA1 hippocampal rat tissue from stratum radiatum in an adult rat, and made a 6 by 6 micron in X and Y by 5 microns in Z, 100 slices of 50 nanometer thick thin sections, and traced out the outlines of all the cellular components. The yellow are dendrites, the green are the axons, and the blue is astrocytic glia. And this 3D reconstruction, as it comes raw off of the tracing software, has a number of flaws in it or distortions in it that need to be corrected in order to turn this from a visually interesting model into with lots of things that you can learn from it just from the morphology of it, into a computationally correct model that's good enough to do computation on. And that was accomplished Making those corrections was accomplished by a graduate student in our group, Justin Kennedy, who's now a postdoc with Ed Boyden's group at MIT. And some of the flaws in the reconstruction are due to the fact that when you dehydrate tissue and fix it for doing EM, there's shrinkage that occurs. And the extracellular space actually shrinks and becomes thinner and more occluded. How do we know? Because the only way to really see the extracellular space is in EM, and the only way to do EM is to fix it and do all this stuff. Well, the way we know that it must be shrinking is that you can do diffusion studies of small molecules in the living intact tissue and track how quickly the particles diffuse through the tissue. And diffusion theory can be used to tell you what the extracellular volume fraction is, meaning the fraction of volume that's in the extracellular space divided by the total volume of the whole structure. And from doing these diffusion studies, it's been learned that that the extracellular volume fraction has to be about 20%. But when you do the 3D reconstruction from the EM and look at it, if you calculate the extracellular volume fraction of this, what you have is about 9% extracellular volume fraction. Plus, there's some additional artifacts that come from doing the reconstruction, which are due to sampling problems. So this is the XY plane, and if you do the 3D reconstruction, and in the XY plane, our XY resolution is about 4 or 5 nanometers. But the Z resolution here, because this was done from serial thin sections, is 50 nanometers. Well, the width of the extracellular space is maybe 20 to 30 nanometers. So 50 nanometers is actually too coarse to be able to fully capture the extracellular space accurately. And what happens is, so if we take the full 3D reconstruction and cut it in the XZ plane, so that now the XY plane is really here, going in this direction, and we've taken the whole thing and flipped it sideways and cut it in this direction. And if we enlarge this little box over here, what you see are places where, so now remember, this is the Z direction, and so the 50 nanometer slices were actually cut in this direction, and I probably should have put a scale bar on it so you could see what 50 nanometers is, maybe with little tick marks along the side. But you can see places where the extracellular volume is, that the width of the extracellular space is very narrow. Some places where it's actually, some places where it's actually occluded completely. This represents a place where these two adjacent compartments are actually intersecting with each other, which is non-physical, obviously, right? And it wasn't due to errors made by the person who traced the outline of this, right? The errors are actually due to what's called aliasing, or sampling error, where there's just not enough density of sampling in order to correctly capture that feature. And so what Justin Kinney did was he wrote an algorithm that corrected and identified these places where there's overlap and eliminated those, and then in addition made it so that we expand the volume of the extracellular space back to its 20% volume fraction. And this is detailed in a paper that should be in the publication list, the method used to do this. Okay, so So now this is part of the step, the complicated step necessary that might be necessary for your own work if you're working from with trying to get geometry that comes from a 3D reconstruction. And if we haven't yet put all these things together into a coherent pipeline, but there's a tool that we're not going to show you because it's kind of beyond the scope of what we can accomplish in just 3 short days. But if anyone's interested in the tools for that pipeline, you can contact us, and we can talk about it offline together. One of the tools is called Vol Rover N, and it's being developed in collaboration with Kristen Harris and Chandra Bajaj at UT Austin. And there was a paper that came out in Neuroinformatics earlier this year on that tool. And we're working to try to couple it into the pipeline to get it so that data can flow more easily into Cell Blender. But we're not quite there yet. But that'll be coming in the next year or so. We'll make that easier to do. And that tool will incorporate all of this massaging of the data to try to recover the data back to its intact in vivo state. And this is just a diagram showing kind of the method that Justin used in order to make these adjustments. He made a finite element model of the polygons and figured out the distance between adjacent vertices on adjacent surfaces, and used an optimization method to create a force that would move the vertices in proportion to the error in the measure, and then iterate on this and converge on a solution. And the result of doing all that was this 3D model. And this is available on YouTube for viewing if you want to later. It was produced by all this cast of characters. And this is the production software used to create this. And what you're going to see are dendrites with their postsynaptic densities, endoplasmic reticulum and mitochondria, and synaptic contacts between axons and dendrites, and astrocytic glia and all the synaptic contacts. This is so now a tour of that 3D reconstruction that Kristen and Justin were able to put together. So this is the star of the show. It's one big apical dendrite out of that volume. And the synaptic contacts areas are shown in this kind of maroon-red color here. And there are 77 spines, dendritic spines sticking out on this particular dendrite. It's a pyramidal neuron. And these are its dance partners, the axons that make contacts with each of those dendritic spines. And this is the astrocyte. It's just one small piece of an astrocytic glial cell that happens to be in this volume. And all these pieces of it belong to one single astrocyte. So all this complicated structure is all just the interdigitated pieces of one astrocyte. And it entwines itself throughout the structure. And if we go inside the dendrite, you'll see we have the endoplasmic reticulum and mitochondria. And there would be plenty of other components in here, like microtubules and microfilaments and ribosomes and endosomes and lysosomes and all kinds of other organelles inside this cell that we haven't yet put into the reconstruction. And here we're going to go up inside the neck of one of the dendritic spines. and into the inside of the spine head, where you can see the inner piece of the postsynaptic density in red there. And if we pop through the head out into the extracellular space again, you can see here's that neck that we just came through. And one of the things Justin was interested in was what's the role of the astrocytic glia in reuptake of neurotransmitter and affecting neurotransmitter. And he noted that some synapses, like this one, don't have much astrocytic glia nearby. But other synapses, like this one here, are partially ensheathed by astrocyte near the synapse. And then around the corner here, there's going to be another example of ensheathment where the astrocyte completely encircles the synaptic contact, and from a certain angle you can look in and see the synapse in there, but this one's completely encircled by astrocyte. And the astrocyte is where the glutamate transporters are that reabsorb the neurotransmitter. And so we thought there would be a big difference in the efficacy of neurotransmission at these 3 different synapses, but he found that that wasn't the case, and I'll show you why in a minute. So, but what looks like empty space is actually jam-packed full of all the other dendrites and axons. And altogether in this whole reconstruction there are 150 pieces of dendrite and 450 pieces of axon, and just that one astrocytic glial process that fully tiles and envelops this 6 by 6 by 5 micron piece. So now if we make everything invisible except for the axons, this is what you see. Except for a few holes here for some of the dendrites, it looks like you could hardly fit anything else. But in fact, this occupies about 30% of the volume. And then the astrocyte occupies about 10% of the volume. So now that's 30% plus 10%, we're up to 40%. And these are the synaptic contacts, and you see that most of them aren't enveloped or nearby the astrocyte. Most of them are kind of floating out in the middle of space there. And then another 40% of the volume is occupied by the dendrites. So now we're up to 80% of the volume. The remaining 10% is the extracellular space, as I said. And so it's interesting that the astrocyte only occupies 10%, while the extracellular space is 20% of the space. And then viewed from the outside, the endoplasmic reticulum and mitochondria are interesting to look at. Notice that the mitochondria are long spaghetti-like structures in dendrites rather than the usual little jelly beans that we're used to seeing. So that's the geometry of the model. The biochemistry of the model and the molecules and reactions of the model also come from the literature and also experiments that myself and my colleagues have performed to measure the kinetic rate constants of some of these things. So we have a kinetic scheme for AMPA receptors, NMDA receptors, voltage-gated calcium channels, the glutamate transporters, the PMCA and NCX pumps that pump calcium, the circuit pumps that pump calcium. So these pump calcium from the cytoplasm back into the extracellular space, and these pump calcium from the cytoplasm into the endoplasmic reticulum. And then there's immobile endogenous calcium-binding proteins that use this reaction scheme. This scheme is also used to model fluorescent dyes like Flo4 and OGB1 in the model so that we can replicate the results of experiments that you can do in the lab. and see whether the fluorescence generated in the model matches fluorescent signals that you observe with a microscope. So this is really a powerful tool because you can use it to replicate experiments you can do in the lab and see whether the result you get is similar to what you see. And if not, then you know you're on the verge of learning something new, like you've got a typo in your model, or there's something going on in the biochemistry that you don't understand, and by probing it further, you can then figure out something new. And then the other calcium binding protein, calbindin. And we know the kinetic rate constants for most of these things, and the ones that we don't, we can adjust by doing some tuning and comparing to experiments and making predictions of what that should be, and then go back and do an experiment to see whether that makes sense or not. And then other experimental constraints on the model are observations, such as after a single spike in an axon, this is the measured release profile of synaptic vesicles from the presynaptic terminal. This is number of quanta released per bouton per millisecond after a single spike in an action potential. And then if I put in a pair of spikes with a certain interpulse interval, I should see this ratio of facilitation occur from the presynaptic terminal. And then we can also put in the kinetics of the calcium sensor in the presynaptic terminal, the snare complex. And some of the kinetics of this have been illuminated, illuminated or brought forth from experiments done on the calyx of Held in the auditory system. And then we adapted this model, this kinetic model, and made adjustments to the kinetic parameters so that we could replicate the shape of this curve. And then, of course, we simulated this with MCell. And you've been hearing about MCell this morning, but I'll just remind you of some of the important features that MCell has. It lets us make realistic 3D reaction diffusion models. And we use rigorously validated and highly optimized stochastic Monte Carlo methods so that we can do these simulations very, very fast, as fast as can be done for stochastic modeling. And you should know, even if you don't understand, I think it's important for you if you're going to do these kinds of simulations more seriously in your career in the longer term, and I hope that you will, that you understand what the methods are that MCell is using, but you can take comfort in knowing that we've rigorously validated these methods for you, and we will continue to. But don't believe us, right? Do it for yourself and prove to yourself that you're getting the result that that makes physical sense. And MCell tracks the Brownian dynamics diffusion of individual particles in 3D volumes and on 2D surfaces. And we can handle arbitrarily complex 3D geometry. And you'll see in a moment what I mean by arbitrarily complex. Well, you've already seen some. And we can handle arbitrarily complex reaction networks that are represented as Markov processes or by network-free rule-based specification of rules. And tomorrow, Jim Faders and his group will show you how the BioNetGen language works and how it factors into MCell simulations. But all these are modeled as discrete event-driven point processes. And what that means is, we'll cover in more detail tomorrow when Jim talks about network-free, rule-based simulations. But what it means is MCell can simulate the rules directly without the need to model the whole reaction network spelled out. It can evaluate the rules on a rule by rule based on a rule by rule based basis. And so even if you have a reaction pathway, this was key to the simulation I'm about to show you. That includes CAM kinase 2. CAM kinase 2 is a dodecameric holoenzyme. It has 12 subunits in it. And each of those subunits can bind calmodulin. And calmodulin itself can be in one of 9 different states, depending on how much calcium it's bound. And so now now calmodulin can bind to each one of the subunits separately of this dodecameric thing. And then each part of the dodecameric thing can be phosphorylated or not phosphorylated at a number of different sites. So altogether, what you end up with is a holoenzyme that where each subunit of each of the 12 subunits can be in one of 40 different states, and there's 12 of these altogether. And so what you have is then 40 to the 12th power different states that the holoenzyme can be in. That's like, I think, like 10 to the 16th or something if you do the math with that. It's like 10 to the 16th. And the number of reactions that you would need to write down to specify all the transitions between all those different states would be about 10 to the 21st reactions. So almost a mole of reactions. You couldn't do it, right? And so if you were doing traditional reaction network-based modeling, you would look for ways to simplify and coarse grain and look for symmetries and things. But even with something as complicated as this, that technique runs out of steam, and you still end up with way too many reactions to write down. So it turns out that you can write down a rule-based specification for that entire mess without any simplifications or approximations in it in about, 80 lines, was it? Or 50 lines, Jim, do you remember? It was like 50 or 80 lines of BioNetGen language for CAMK2. A complete, yeah, but it was on the order of 50 to 80 lines of rules. Yeah, a complete specification without any approximations. Now, if you were, well, but I'll leave it at that because we'll cover this again. Jim will cover the horror of combinatorial explosion tomorrow in more detail. But CAMK2 is kind of a poster child for that kind of thing. Anyway, so and we can do all this with this high-level model description language that. The way we get all this capability and flexibility is because we have this model description language. And Marcus gave a kind of a brief tour of that this morning. But because we have this new model building environment, CellBlender, you don't have to worry too much about the model description language if you don't want to, depending on the needs of your simulation that you want to do. Okay, so Marcus already covered all that. All right. So I'm going to give you. So here's part of the model that I'm going to show you, the presynaptic side of the model that I'm about to show you. Previously, we had published a paper on the presynaptic part of the model using this very simplified, what we call a canonical model based on kind of cuboid-shaped things. And we published this in a PLOS Comp Bio paper in 2010, and then a follow-up to that paper in PNAS in 2012. In which we have docked vesicles and snare complexes, a calcium sensor, and a cluster of voltage-gated calcium channels, and then calbindin in the cytoplasm, and PMCA pumps on the extracellular membrane. All of this is located right in here. And then what we've done now that I'm about to show you is reinstantiate this whole model. So this is one of the powerful things you can do in this kind of modeling is once you have a model that has all the biochemistry in it and mapped onto some kind of geometry, you can replace all that geometry with new geometry, and as long as the names of objects and the names of regions in it are correspond to each other, then you can just do a drop-in replacement of this geometry with this geometry, and all the biochemistry maps onto it. And so what you see here is the same system in which pretty much all the same thing, except we've added a few more. Now we have endoplasmic reticulum in it with the circuit pumps on the surface of the endoplasmic reticulum, and we've added IP3 receptors and metabotropic glutamate receptors and phospholipase C, and the calcium sensors again, and docked vesicles, the cluster of voltage-gated calcium channels, calcium floating around in the cytoplasm. We've got calbindin floating around, PMCA pumps, the circuit pump, right? And this is what you're seeing here is a magnified view of this little portion here, and this bouton. This is an axon, and these swellings are called varicosities or the boutons. And you'll notice that this axon has a bouton here and another bouton there, and there's another synapse here. And this bouton doesn't have a mitochondrion in it, but this one over here does. And so you, from the 3D reconstruction, you get all this additional richness and complexity that makes you wonder what it's all about. And by doing these simulations, you might be able to make some predictions about what it's all about that might be testable in experiments. Okay, so now if we put it all in motion, what you're seeing here is that same 3D reconstruction that you saw in the YouTube video, but now we've instead of just being the structure, we've now brought it all to life again. So here's that axon I just showed you, and the particles diffusing around in the cytoplasm. As we zoom in closer, you can see these are those are the calcium ions. As we zoom in closer, you can see the synaptic vesicles. Here's a cluster of voltage-gated calcium channels, and as we get closer, the axon's going to turn a brighter shade of green, indicating that an action potential is happening. The voltage-gated calcium channels open. Calcium starts to flow into the presynaptic terminal. The little black particles are calbindin. They bind some of the calcium, but still the calcium can build up and bind to the snare complexes over here. And when one of them binds enough calcium, it releases neurotransmitter. That's 2,000 molecules of glutamate get released, and they diffuse through the extracellular space. And it looks like it's empty here, but it's all jam-packed full of that other stuff. It's invisible. The calcium, the neurotransmitter diffuses and binds to glutamate transporters on the astrocyte. They turn red. And then on the postsynaptic side, the glutamate bound to AMPA receptors that are green here and NMDA receptors in blue. And when an NMDA receptor flickers to the open state, it lets in a little burst of calcium. But the NMDA receptors have a magnesium block. At hyperpolarized potentials like here, at the resting potential, the block is fairly heavy, but not absolute, so some calcium come in. But when a backpropagating action potential arrives, the NMDA receptors can open more efficiently and let in a lot more calcium. And the calcium binds to calmodulin, which are the little magenta ones floating around, and there was another release of neurotransmitter. I'll talk about that in a second. The calmodulin can bind to CAM kinase 2, which are the big blue, light blue, and dark blue blocks. And so you can, if you have a pairing of presynaptic activation and a backpropagating action potential, you can get this large influx of calcium that leads to a large activation of CAM kinase 2, and eventually, at longer time scales, a change in the efficacy of the synapse, where it gets stronger and has a bigger EPSP and a higher release probability on future events. So this was a hand-picked simulation in which there was a release of neurotransmitter. 80% of the time when there's an action potential, there's no release of transmitter. 100% of the time when there's an action potential, you have activation of the voltage-gated calcium channels. 100% of the time you have this influx of calcium. 100% of the time it binds to the snare complexes, but only 20% of the time is that binding to the snare complexes